Um, Hello everyone. Good afternoon. This is Mohammad Mahfuzur Rahaman, PhD student from Computer Science Department of the University of Central Florida. Today, I'm going to discuss our newly developed tool, RNAMotifComp, which analyzes the RNA 3D structural motifs and identifies structurally similar motif families. Before starting to discuss the tool, let's review the concepts of RNA 3D structures and the definition of structural motifs. Although the RNA molecules are single-stranded, they have the ability to fold into complex 3D structures to exert their function. This folding is driven by their chemical structures, which include base pairing, stacking interactions, and loop formation. Therefore, an RNA chain can be differentiated into two regions, one of which is helical regions, and the other is loop regions. These are the helical regions, and these are the loop regions. Helical regions refer to the region in which RNA folds into a double-stranded helix, and loop regions are the unpaired segments of RNA molecules that connect the helical regions. The loop regions are characterized by the absence of base pairing interactions and can have diverse lengths, sequences, and conformations. They provide flexibility and structural diversity to RNA molecules, allowing them to adopt complex three-dimensional structures. Based on the 3D structures, the loop regions can also be categorized into um, hairpin loops, internal loops, and multi-loops. They play critical roles in various RNA functions, including RNA protein interactions, enzymatic activity, ligand binding, and regulatory mechanisms. RNA 3D structural motifs are recurring patterns or arrangements of RNA secondary structure elements that contribute to the overall three-dimensional architecture of RNA molecules. Based on their structural similarities and base interactions, these recurring segments are organized into several structural motif families. For the example RNA chain depicted here, we found 10 well-studied regions, which are highlighted here. As mentioned before, the red ones are named as Kington motifs, the green ones are named as sarsenicin, and so on. So it is evident from this picture that the instances from the same motif family are supposed to have similar structures, while the instances from different families are supposed to be structurally different. So detailed analysis has been done on the various variations of such structural motif families, and it was proposed to subdivide each family into several subfamilies based on their structures. And the work has been published as RNA motif contrast. Here you can see the example of the reverse Kington motif family. Uh, the RNA motif contrast actually, by analyzing their structures, the 3D structures, it's subdivided into three different subfamilies, and you can see the differences from this figure here. Like all the subfamilies, they are not same, but they are of the same motif family, which is reverse Kington. So we have done further analysis of these motif families and discovered structural similarity across motif families. So for example, the red instance from the Kington, here the red one is from Kington motif family, and the yellow instance from Eloop family, they form different shapes, but when we look into their base interactions, a significant matches with the other. On the other hand, in the second example, the yellow instance from the Eloop motif family and the magenta instance from the tandem shear motif family, they have a similar shape, which is visible in the superimposition figure here. So these two examples prove that there exists structural similarity across motif families from both their interaction and shapes point of view. So we have used the dataset of 360 internal loop motifs from 11 well-known families to analyze their similarities and designed RNAMotifComp to identify similar families based on some particular criteria. So the major problem of the RNA structural motifs are they can form similar shapes with different base interactions. Also, they can form different shapes with similar base interactions. Therefore, both interaction-based and coordinate-based alignment algorithms face difficulties in identifying new instances of a motif family. It is important to note that due to the variations inside a motif family, the average RMSD of all aligned instances of the same motif family will not help us to identify the differences among motif families, because due to the similarities across motif families, the average RMSD of the aligned instances might become small enough to be comparable to the previously mentioned RMSD. Here's the workflow of RNAMotifComp to collect data and analyze structural motifs to identify similar families. So RNAMotifComp automatically collects. So in this example, like we have used the process for 5 motif instances from 2 different motif families. So RNAMotifComp automatically collects specific PDB files from the PDB database and separates the PDB coordinates for each location into new file. The annotations of each PDB file are collected from 2 different sources. One is from DSSR and the other one is from FR3D. Then the 2 annotations are merged together for better annotation data. So then we apply an interaction-based alignment algorithm to find the interfamily pairwise alignment of all the motif instances. And based on some preset criteria, the better aligned instances across different families are identified, and percentage of participating instances is calculated. So here the green ones are the percentage we have calculated, because the red ones are out of threshold. So this criteria include a better alignment score, alignment greater or equal to each family specific alignment length threshold, and a lower RMSD value. So in this example, at least 50% of instances of each family are getting similar to one another. And similarly, we repeat the same procedure with coordinate-based alignment tool to find the coordinate-based similarities across motif families. And that's how we actually generate each edge between 2 motif families to generate a similarity graph. So this is the summary of all our inputs. As mentioned earlier, we have used 360 motif instances from 11 well-known motif families. We used 2 different annotation tools for better accuracy. We used 2 different alignment tools two different annotation tools for better accuracy, and 2 different alignment tools to show the similarity from 2 different points of view. Similarity criteria are better alignment score, lower RMSD, and a significant percentage of participating instances from both sides. The output we get from RNAMotifComp is a similarity graph showing similar motif families along with other necessary information. And for the motif family names, in further slides, we have used these short forms, and these are the 11 names of well-known motif families that we have used for our experiment. So this is the similarity graph we have found from the RNAMotifComp using the interaction-based alignment tool. So it shows that eLoop and King-term motif families have interaction-based similar instances. And similarly, eLoop and tandem shear motif families share base pairing and stacking interactions. From the age level, at least 34% of instances from each family are similar from their interaction point of view. We have generated the consensus secondary structure of these participating instances to show the authenticity of our claim from this figure. Although King-turn has a bulge here, you can see, but the significant portion of their base pairing interaction matching. Also, they might have similar stackings, which is not shown here to keep the figure simple. We have also generated the consensus secondary structure from the participating instances of eLoop and tandem shear motif families, which also supports our finding. Like the gray one, the AG base pair, that's like less frequently occurred, but most frequently occurred base pairs, which are there, like the 2 different AG base pairs, which is common for both the motif families. Now the next slide, it shows the similarity graph using a coordinate-based alignment tool. Like instead of the interaction-based, we have used the coordinate-based tool here to show the instances. Like it shows instances from eLoop and tandem shear have coordinate-based similarities. Moreover, RNAMotifComp found C-loop tetraloop receptor, eLoop hook turn, hook turn tandem shear, and hook turn tetraloop receptor as similar motif family pairs, having a good amount of participating instances. The superimposition images of the participating instances from E-loop and tandem shear motif families are shown here, which visually proves our findings. So, um, so the summary. So here, this actually is a side-by-side image of the superimposed images for better understanding. Although the instances are from two different families, they kind of look very close based on their shape. And here is the summary of our findings, which is the similarities among the well-known motif families. Based on our analysis, Kington and E-loop share a good amount of their base interactions, and so they are similar based on interaction. E-loop, hook turn, tetraloop receptor, C-loop, hook turn, tetraloop receptor, and hook turn tandem shear. They have similar shapes. E-loop and tandem shear have both interaction-based and coordinate-based similarities with sursiniticin, reverse Kington, and L1 complex. Rope sling and T-loop do not have any significant similarities based on either interaction or coordinates. So to verify our findings, we compared our result with RNA-motif contrast. RNA-motif contrast takes a group of motif instances. This is an existing tool. So it takes a group of motif instances and as input and considers them as one motif family. Then based on their structural variations, it distributes them into several subfamilies. We provided all motif instances here from different families as one family. Theoretically, RNA-motif contrast is supposed to separate the instances from different families into separate subfamilies due to their variation in structure. If it cannot differentiate the instances from different families, then they must have similarities to some extent. So to do so, we actually followed these steps. First, we took all the motif instances and removed their motif family annotation. Then provide these to RNA-motif contrast as input. After getting the output from RNA-motif contrast, we re-annotated the motif instances with the original motif family data. So here is the output of RNA-motif contrast. Similar families claimed by RNAMotifComp are highlighted in orange color, and you can see the result here. Why RNAMotifComp did not claim the other families as similar, although like they are in similar family based on RNA-motif contrast. So the reason is they did not have significant number of participating instances to claim the families as similar. Like the threshold we have set, like 20% or 30%. So that actually didn't meet that criteria. So that's why, although they have similar instances, but that doesn't pass the threshold. So that's why the RNAMotifComp did not predict them as a similar motif family. So to evaluate the utilities of this tool, RNAMotifComp, uh, we implemented two Gaussian knife bias classifiers. One is interaction based and the other one is coordinate based. Our goal was to show the Gaussian knife bias classifiers perform better with the motif families with unique structures, and the performance degrades if the provided motif families have similarities in their structures. Alignment score, alignment length, RMSD, etc. are the features we have used while implementing the Gaussian knife bias classifiers. So here's the result from Gaussian knife bias classifiers. And according to this table, our classifiers perform better with the Sarsinicin motif instances in both interaction-based and coordinate-based classification compared to other motif families having structural similarities. So we intentionally choose the motif families whichever has similarities with other, and one of them we choose which doesn't have similarity, like which is unique, which is Sarsinicin. So from there we can see like the Sarsinicin is getting better accuracy compared to the other motif families which actually share their shape or base interactions. So to conclude, we can say RNAMotifComp can successfully identify similar instances across motif families. The similarity threshold values are adjustable through command line parameters, so the user can change them according to their requirements. As it is clear that different motif families might have similar instances, it is possible that the source of this could be some errors or misannotations. So re-annotating those known motif instances could be needed. The annotation inaccuracy could be one potential source of interaction-based similarity. So finding those annotation errors could be one possible research scope. Along with these, RNAMotifComp can be utilized to measure the quality of motif clusters, which in turn will be helpful to improve clustering results. And that's all for my presentation today. And I'd like to thank my supervisor and my collaborator. And I'd also like to thank NIH for the funding. The RNAMotifComp will be available in this GitHub repository. And if you have any question, you can ask now, or you can leave it in the GitHub repository, or you can contact personally. That's all for today. Thank you so much for hearing my presentation. And yeah, that's all. So yeah, if you have any questions, please.
Any questions?
If not, I can stop the sharing then, right?
Can you hold on 1 second?
Yeah, yeah, sure.
You can just speak.
Thanks for your presentation. I was wondering if you found, if you investigated whether similar motifs are more likely to be bound by the same RNA binding proteins.
Okay, yeah, we actually are currently focusing on the similarities, and then whenever we'll dig into it, like the reason for this, so then we might be able to get the sources of that one. So, like right at this moment, we just found the similarities, and we are trying to like study more on this one.
Thanks.
Thank you.